I just want to show how you can take the basic ideas that I've shown you and expand them up. So this was our first pathway we produced back in 2008. My long-suffering student, Sabir Raza, was able to take various bits of ideas around pathway notation and begin to build this diagram. So this is a macrophage, these are the TLR systems, this is apoptosis, this is NF-kappa B, these are the interferon pathways. So the pathway we have just drawn is somewhere up there. Now, when we started this diagram, we didn't know what we were doing. We didn't even know what tools we wanted to use. We didn't know what tools were available. And one of the things about you learn as you go through this pathway modeling is you not only learn what you think you know, you realize what you don't know. So by actually drawing something out and spelling it out in detail, you realize actually that NF-kappa B system, that's quite a small I mean, it's a lot more complicated than that. Oh, and we haven't included a whole range of other signaling pathways which we could draw on this thing. So by actually modeling out systems that you're interested in, there's not only a way of defining what you do know, but also identifying what you don't know, gaps in your knowledge. So this was our first effort. We were quite proud of it at the time. I look back on it now and it's not so great, okay? But that's the evolution of thinking and evolution of approaches. And in fact, the tools have actually improved since then. So after finishing this diagram, we had another cohort of students when I worked down at Little France, and we gave them all. So the basic way that we have been evolving this notation system over the years has been taking people like yourselves and saying, you know, you have, so MSc students have projects sometimes. In this case, the MSc was in pathway medicine. And so we actually gave them a piece of biology and told them to take any resource that they wanted and to come back to us and draw that biology. And this is a very iterative process whereby we would meet maybe once every week or 2 weeks, and they would come back and say, I've drawn this bit, but I don't really understand how to draw this. And so we would sometimes battle that out, or it would be a simple solution I hadn't shown them before. And in such a way, we were able to build up pathways. So our NF-kappa B system here, which was very naive, turned out to be this big when we actually drew it out in detail. Rather than 2 transcription factors, I think there was something like 14 that we identified within the literature. This was all the non-TLR systems, so these are systems by which a macrophage identifies a pathogen. There are lots of different types of ways that a macrophage may do this. We'd only include the classical toll-like receptor signaling pathways. We hadn't included these non-TLR systems, and that was that diagram. We redrew the TLR system because we thought it was a lot better, and then we ended up drawing a whole range of other pathways in MAP kinase signaling pathways, which are linked to this pathway, but we hadn't defined here earlier on. This was an attempt to draw antigen presentation. So again, one of the things which is obviously a macrophage does is it digests bacteria and viruses, chews them up, and it presents their antigens on the surface. So this is the proteasome. And actually, when we went in this, I always thought there was a proteasome. Yeah, you talk about proteasome. Turns out there's 5 different types of proteasomes, and 3 of which are devoted to presenting antigens. Okay, you don't know this stuff until you read and present it. And then essentially, this is the systems by which they present the class 1 or class 2 antigens. This is lipid metabolism drawn out in some detail. And this is a very brave attempt at modeling the cell cycle. So these are all diagrams have been done by people like yourselves. No more experience than I've given you today, but with essentially this iterative process. So what I'm trying to emphasize is that actually once you get your eye in on this, that you can draw very complicated diagrams. You can actually digest the literature into a diagram. And in the end, we put all of this together. I'll come back to that in a second. Where's it gone? That was the one. So we ended up drawing this diagram, which was once you'd actually had all of these parts, we then joined all of these parts together to make this diagram. The point thing about this diagram is that there's nearly 1,000 papers represented there. Okay. Nice thing is I haven't had to read them all. I've read a few of them, but actually it is a way that you can communicate information by a diagram And as we've seen, we can add various bits of information to that diagram. So it isn't just a diagram, it becomes an information resource. And often now, even now when I'm reading a bit of new macrophage biology, I'll try and center where I am using this diagram because this represents such a wealth of information. So just one other thing to say, and we'll come on to this in a moment. I'll come back to here. So we've put together this website now. I'll probably show it you live rather than on the slide, called Virtually Immune. And so we basically, as I say, for nigh on 7 or 8 years now, we've each year, not every year, but most years, we've been working with cohorts of students in defining these pathways, undergraduates and postgraduates. And what do you do with all these diagrams? Well, they're not kind of publishable in their own right, but they are quite nice. So we've put them all together on this website called Virtually Immune. And if I go here, you can see it here. And so, for instance, we can go, there's a kind of wee tutorial there. So we have this idea about reading the literature. That's that little text that you've just done, and it identifies not only the components, interactions, and compartments. And we show this system there, and there's the diagram you've just drawn there. And then what we're going to go on, and this is what we're going to cover, is how we actually turn this into a computational model. And I'll come back to that in a moment. But importantly, within this website here, we have a gallery of pictures, which we'll come back to in a second. But essentially, within this system, we have a whole range of pathways listed. Some of those I've just shown you now. And you go onto an individual pathway, you can download the file, and you can launch it just within Wired through a WebStart type of system. So again, if you're interested in this, there's a resource there that shows you better diagrams. There's a number of these diagrams in your own folder there, and here's just one loaded here. And so that kind of gives you a detail of the kind of thing that you can do if you spend enough time and effort. But what the point is that it summarizes an awful amount of information, and there is no other resource for this information. So I kind of feel quite knowledgeable a lot about these systems, and the reason I feel knowledge about them is because I've actually interacted with the person. So sometimes actually the most important thing about making a pathway is the process, the thing that you learn in the process of actually constructing it. The end product is nice, don't get me wrong, but that process of actually having to think logically doesn't allow you to read things and just glibly go, ah, yeah, I've read it. Okay, you have to pull out the information from those. One thing that also sort of just came up over the break is this idea, this diagram, you have to, when you get modeling on this scale, it's no longer something that can be presented on a piece of paper. It's something that is designed to work in a computer, that you can zoom in, because if you try and fit that within an A4 sheet of paper, it's no longer readable. It is a diagram that is meant to be read on a computer. And also, I think the other thing to mention is that actually how you lay out things, the order, the systematic way. So the information would be the same if I jumbled all of that up, but it would be no longer readable. So if I was to take this layout algorithm and just say, show it as an organic layout, it's exactly the same information, but of course, it's completely unreadable now. So what you're trying to do when you make a pathway is actually not only render the information of nodes and edges, but render it in a way that a human can read it and somebody else can come along and read it as information afterwards.